and I think we'll get started. I'm going to see what I do. I'm not great at sharing screens and various things, but Jay and I came up with a way to go over this thing. It was both from industry, from Pfizer, and let me introduce Jay that you can see on the at least I can see him. Hopefully you can. And Jay is the anti-infective global medical lead at Pfizer, and since they are in the antifungal field and are continue to be in the antifungal field, he is a big player on this. And what we decided to do was these breakouts. Each one of the kind of industry people had a breakout with an MSG member, and so we are together, both Jay and I, and we didn't know exactly how to do this because the issue was around, well, okay, so Pfizer has the new drug, phosphomonogipex, or they're in a study of that, as they bought that from Amplex. Actually, Jay, in his position at Pfizer, really can't talk about phosphomonogipex. That's in clinical trials, various things, and doesn't really have anything to say specifically about that. And I said, well, okay, fine. What are we going to do? So what I'm going to do is I'm just going to mention a little bit myself, since I'm not in industry. I'm at Duke University, just mention a little bit about it. And then what Jay had done was he said, well, there's some questions I want to ask of you and the groups and stuff like that. And so he listed those. And I said, well, Jay, if you're going to ask questions of me, then I'm going to ask some questions of you. And so we got about 4 questions from each group to kind of go back and forth on this. And so you may have come to think a little bit about some of the drugs that Pfizer has, and particularly phosphomonogipex. And you're probably not going to hear too much about that. But let me just start with saying, again, that particular drug is in clinical trials. Andre was very good at doing a GTW inhibitor. That's an inositol acylase that works on some of these GPI anchored proteins. It is very, very powerful target and in the cell wall, and it's been around for a long time. This drug was back with ESI years and years ago, and so it's broad spectrum, as was mentioned. It has had studies done in refractory candidiasis, small studies, and is now in studies with refractory mold infection. So it goes across both yeast and mold infections, and then more studies are coming. So I think this drug, which has an oral bioavailability, mainly because it's a pro-drug, but it also potentially could be given IV. I would like to say my experience with these compounds in this class is very, very impressive. And in fact, this whole class of drugs that have made other types of congeners off of this. And for us, we've seen some congeners for certain fungal stuff, for certain fungal species, can be absolutely incredible. And so Jay asked one of the questions, which we'll come to in just a moment. Is there a place for narrow effective medications? In other words, not broad spectrum, but narrow. And I don't know about that, but I do know that this class of drugs can be manipulated to make them extraordinarily potent against certain species. So that is a background. What I'm going to try to do is turn this on some global share screen to see if that works. Sorry, we're going to do it without sharing. There were 4 questions, and I'm going to give this first one to myself, which is questions from Jay. And the first question that Jay had out there was, what value would autopsy studies have in better understanding the current situation of missed invasive fungal infections and better raise the bar for suspecting invasive fungal infections? Now, again, any of you that want to weigh in here, please just add or go into the box and stuff like that to add your opinions. But from my standpoint, Jay, I think autopsy studies would be good at this. They could help be more precise. However, on a practical basis, the amount of autopsies done, at least in the United States, is extraordinarily low. And now in the arena of COVID, it becomes even more uncommon and stuff like that. So I think the best studies you have are some of the ones out of MD Anderson that were done 5-10 years ago when they went back and looked at this. And I think they were revealing in that they were seeing many more of the Candida infections than they thought. Everybody was focused on molds, and I think they blew away that type of thing, and they're back recircling back to Candida infections. So I think it would be good to know that thing, to be more precise of that, but to get these autopsies and get them done, the cost of them, to get patients to say they're going to do these autopsies, and particularly a lot of these infections are end of life type situations, and the families are past that type of thing. They don't want to do, and we do have a lot of data on scanning and stuff like that of what patients may have. But I just think your question is important, but I just don't see it becoming a common thing that people are going to get. Maybe other countries can do this thing better, but within the United States, autopsy studies, I think would be revealing. I think they'd be uncommon to get done. Second question,
John,
Jay,
do
yes.
you want me to try and share the screen of this other questions the same as you sent me once? I can do that.
Okay.
And tell me if you can see the screen.
Oh, yeah, there's Jay's questions. Yeah, you got them. Okay, I'm glad you can do them. I'm glad I sent them to you, Jay. Okay, so that was the first one. Second question of Jay's was, what can industry do to encourage or support better identification of targets for diagnosis and treatment of fungal infections? Now, I thought about that. I mean, Jay and the industry trying to do things, it'd be always nice to be able to have research grants or support systems to get our ideas out there, to get them off the ground and stuff like that, and have industry potentially support that. But I actually think that as I thought about this and have done this a couple times recently is, and this is a tricky business, but I really think it's kind of important. And what I think if industry could support those and the government, I believe we need better centers of excellence in fungal infections. And I'm talking about a full service type situation, from the clinical aspects, the basic science aspects, and the research aspects. We need coordination, not just in one country, but internationally. And I think we need to compete and have these type of places that become central areas for new ideas, the ability to have coordinators and stuff like that to do clinical trials. We are struggling to even do clinical trials for approved drugs, or the ability to get approved drugs, letting on ideas of our own trying to figure out how we can tweak these things diagnostically and therapeutically. And so I think it's a big ask, but I do think that sometime there needs to be a change, just like there was cancer centers and stuff like that around the country in the US. I think there needs to be centers of excellence well supported by industry and governments to really be the initiators of diagnostic and treatment type things and ideas and abilities to get things done. It is extraordinarily difficult in this day and age to actually do clinical trials with all the regulations that we have and all the expenses of coordinators and getting out on the wards and at the bedside. So the next question I have that Jay gave to me was, what is the role of antifungal prophylaxis in the ICU for preventing IFIs, if at all? Well, Jay, I think that this has been around for a quite long period of time, and people have tried various types of ways to do it. I think one of the important points that one has to realize with ICUs is that many of them are not created equal, and the patients that go in there are not equal. And therefore, it is so variable sometimes that it's hard to really come up with an antifungal prophylaxis that could show anything, because some of the patients may have extraordinarily low risk factors and others have extraordinarily high risk factors. So if you were to do any type of antifungal prophylaxis, you're going to have to stratify patients on risk. When you start doing that, then you have a much more difficult time to get the patients into the studies, et cetera, et cetera. And the concern always is to expose these patients to antifungal agents for long periods of time in these really, really high risk patients is drug resistance. So my suspicion is that antifungal prophylaxis in the ICUs will not be as productive and have struggled for long times to do. Where I do think that I won't be too cynical on this thing. I think we need to be in the ICUs and understanding what we're doing, because we at our place and many places spread a lot of antifungals over these, quotes, high risk patients. I think we've taught so long with the antibiotics that we've used that fungal infections are also right there. And we need to get to those early infected type things. And so at a drop of a hat in these ICUs, they will be put on antifungal therapies, mainly a lot of it because drugs like the echinocandins are like almost given normal saline. They have very low toxicity and very low drug interactions, and at the drop of a hat, they will be started. That said, I think as was talked about in stewardship and every place else talking about this, I honestly think that preemptive type therapies or targeted type therapies still need to be pushed really hard in these situations. We need to go back and take some of the diagnostics that we have and actually use them in a kind of preemptive strategy type situation and see if we can actually end up diagnose these infections. Or on the other side of it, I think sometimes we push so hard on the positive predictive value of these diagnostics, when in fact, some of them, it's the negative predictive value. We got to stop these drugs. It's like Jose said, I mean, we get these drugs on forever. They get on, put on, and they don't cause any problems, so they just stay there for days and days and days, when in fact, the emperor has no clothes. That I think there's some of these things that you could actually use in strategies, particularly the glucans and stuff like that, where you could stop the drugs after you've already started them. Then finally, the question that he brought up was, should the future of antifungals focus on narrow effective medications or broad spectrum coverage? And I thought about that a little bit, and I think that the easiest way to go on this thing, the easiest way to go is I want the broadest spectrum antifungal I can get, because I don't know what it is. The diagnostics are still suspect. Many of the patients we have, we can't, tissue is the issue, but we can't get the tissue. And so I know how the clinicians think. They want to go to this thing, get to it early. They want to cover everything. They do not want to wake up in the next morning and have something say, oh, that positive blood culture, you'd had them on the wrong antifungal. So it's the same thing with antibacterials. These are wish lists. These are things that we want to do. I think we have to say that the best thing would be actually broad coverage. The other thing that broad coverage also gives you, Jay, I think, is the fact that some of these things, and particularly new development drugs, and we've done this in the past, so there's precedent for doing this thing, is some of them may end up being used as prophylactic agents in high-risk patient populations. And there, you don't want to have too many leakiness to these things. One of the big advantages you've got with the antifungals is that there's no, not I'm aware of, any passage of transposon mediated or plasma mediated drug resistance like there are with bacteria. Surely you can select for drug resistance and stuff, but it gives you the ability potentially not to have the kind of overgrowth that you might have with the bacteria. So I think broad spectrum is attractive. That said, in the same discussion, we're looking at Arulafilm or the F2G drug. And I think, again, that's a fairly narrow spectrum, or at least a spectrum of mold, and there's not yeast activity. And it would be quite interesting to see as we go along the line how drugs like that might not be the best prophylactic agent, but might be very attractive for very focused type things. And if our diagnostics are good, it may help. The other thing about the narrow spectrum ones is, again, something that we need to think about the future. It's not easy for industry to do this, but for us to put together in kind of combination therapies. If these drugs are very, very potent and they work on certain different mechanisms of action, we still need to think how we could do combination therapy, because in the end, I suspect many of these drugs in the future that could get the organism cleared away as fast as it could will maybe require combination therapy hitting multiple sites, just as one has done sometimes with the anti-neoplastic drugs. So those are my questions, and if anybody wants to ask anything about those or add to those things, please either get on or put it in the chat. And
can I follow up on the last point there? And
sure.
Okay, if you have a wish to run one study with a combination, what would that be?
If I had one wish of one combination to do,
yeah.
Whoa, that's a tricky business. But I think what it would be in a mold infection with an echinocandin and an azole compound. And here's my argument. And you got me unplugged pretty quickly here. The echinocandin might actually help in those molds by slowing down hyphal tip growth. And sometimes you're in the vessels and they're going, you really want to stop it right in its tracks. You may not kill it with that echinocandin, but you could stop it from pushing forward, and then use the azole compound or another compound to actually kill the organism. So I think that's my first thought, is the echinocandin azole. But again, I need to think more about that. I may change my mind tomorrow.
Thank you very much.
Anybody have any other questions before we go to questions that I'm going to ask Jay? That he gave me some softball answers, or the ability to do softball answers. I'm going to give you some kind of tough things a little bit, and that's why I think I'll read them myself, and then he can attack them. So these are my questions back to him, and I'm going to start with the first one, so he's ready to kind of think through this thing and come on. What if I said the most important need today in antifungal therapy is rapid, safe kill of fungi? For instance, drugs with rapid action and only given for a few days for success. I had no idea what Joe was going to talk on. I had no idea. So this was completely just me sitting down thinking. Does industry consider this too risky and not financially viable? In other words, if we only use it for a day or two or three, is that just not the way that that it would be helpful to companies to sell this thing, or they would have to use such a high price in these drugs?
Not at all. Not at all. A very fast kill, very narrow spectrum can be very viable. Yes, I don't want to say disagree, but it did not reflect my view when, gentlemen, I please forgive me if I don't remember who it was in the beginning.
It was me. I'm here.
That was you. Yeah, I apologize. I do not think. I mean, it might look on the face of it that's not financially viable. Now, I am not a commercial person or marketing. I am a medical person at Pfizer, and I don't know if you know, at least at Pfizer, we have a wall between the commercial organization and the medical organization. There is definitely, and it could be very interesting. We recognize internally that efficacy, even with the best drugs available, including Pfizer's drugs, is still not acceptable. There's still too much high mortality. So if something works fast, it will be of interest.
Well, thanks, Jay. I agree completely with that. And Mauricio had that statement. I think that is not a real issue, because one of the issues here is if those drugs are fast, effective, they have several advantages. Number one is the underlying disease can be treated a lot better if you get out of there and don't stay around. And two is I think that killing the fungus rapidly, outcomes are going to be better, and that's an important issue also. So, Mauricio, to you and to the others, you're somebody from industry, at least on the clinical side, saying, I think that's a faulty statement that that rapid action, killing fast, wouldn't eventually have major implications to the management of these patients. In fact, make their total cost be even less by better outcomes and less resources put into other things.
I totally agree with you. I think my mistake in that case was to talk to a businessman in the wrong way and present the way that the drug is presented in a way of how the action of the drug works instead of how the drug would be administered, which is a different concept.
Right.
You know, the investor already think about the time of the killing as the time of administration, which is totally different. So I should have presented in a way that the drug benefit, the patient will benefit from this one. The patient may be willing to pay more, or the company would be willing to sell the drug at a much higher cost. So, you know, I think you have to take into consideration how you will present the data to an investor. Is a different way how you present the data to a scientist.
One comment, if I may, just quickly, Pfizer already has a drug that is administered once, and that is diflucan for vulvovaginal candidiasis. So we have some experience with that, and I can't speak for marketing and financial view exactly, but I do know that there is definitely a way forward for something like that.
Thanks, Jay. I'm going to go to the third one here because I say we've got 5 minutes now. It's 4 minutes or so. So
Okay.
We're talking a long time here. I had a question from an industry standpoint. Does an antifungal need $1 billion appeal in sales? And that's kind of like the silo type situation because that's what they see, et cetera, et cetera. But on the other hand, what if you could show that it could save the healthcare system billions of dollars for being very effective, very efficient, and stuff like that? Is that the way we should be talking to industry, to healthcare systems, et cetera, et cetera? And it may not just be that these drugs are going to save, or they're going to make a whole mess of money directly, but indirectly they could have major impact on healthcare systems.
Yes, no, the answer simply is no, it doesn't have to bring $1 billion. And without divulging things within the organization and how Pfizer is huge, and in a huge organization, everything has to have a huge benefit in order to show. But Pfizer has addressed that. And so let me just get to the point where there is, well, I'm sure there is a minimum, but you can assume that there is no minimum when you present an idea. So, for example, like that, back again to the same first question, I don't think you need to worry about that much of a reward for someone like Pfizer to invest in it.
Great. And let me ask this other question, because we could get knocked off all of a sudden, just like this. Yeah. Fungi are generally not passed from person to person, except maybe for vaginal candidiasis and some dermatophytes. Thus, IFIs have little curb appeal on Main Street. How do we lose that? And how does Pfizer look at that, or how should we look at it on an advocacy point? That just doesn't make it like COVID, for instance.
There's more to being, working, having medications than just the individual. Remember that Pfizer also prides itself to have an impact on overall and not just one particular drug. So there is more to the answer than I can say.
Yeah. Jay, I'm losing you a little bit, but I understand what you're saying. For advocacy, we need to make sure that it's not just that IFI, it's the whole process of what that IFI means to underlying diseases and stuff like that. It's a whole system.